All right, we're seeing your screen.
Okay, thanks, Chip. You see a title slide right now?
Yes, we do.
Okay, good. Yeah, so thanks for the introduction and thanks for joining. This talk will be about low recruitment, particularly in the South Atlantic region for a number of snapper grouper species that we've done stock assessments on, but also some other species that are exhibiting this low recruitment that we haven't done stock assessments on. Let's see. It's been a big team that's worked on this, and I'll be talking first, and then Anna's going to take over midway through the presentation to present some of the other work. But Kevin Craig has also been a big contributor, and we've had a number of other contributors, more than are listed here, but I did want to call attention to Nate Batchelor, Nikolai Klebansky, Brendan Rundy, Caitlin Wade, and Eric Williams, who have helped a lot on this project. But before I go on, I want to define recruitment because in fishery science, it has a number of different definitions. But what we're talking about here is recruitment of young fish into the population. And this process is arguably the main driver of population dynamics for any particular stock. And so as such, it's very important for how managers define what is a sustainable harvest. So this has some big implications for not just the population dynamics, but also the fisheries. And now I would like to advance my slide, but it's not responding. There we go. Are you seeing a second slide now?
Yes, we are.
Okay, thanks. So we first started noticing this pattern of low recruitment from doing stock assessments, and a number of species exhibited low recruitment in the recent years, recent meaning last 10 to 15 years. And those were black sea bass, gag, scamp, and red grouper, red porgy, and snowy grouper. But we also saw evidence of low recruitment from fishery independent data, particularly the Southeast Reef Fish Survey, SURFs, and the reports on species that aren't necessarily targeted by the fisheries, such as bank sea bass, knob porgy, sand perch, and scup. And the evidence has been documented in several peer-reviewed publications, one on scamp, one on red porgy, and then one that examined multiple species from stock assessments. And so when I say what is this pattern that we're seeing from the stock assessments, this shows that several of the species, the red porgy, black sea bass, red grouper, gag, snowy grouper, and scamp. And what this is showing over time is the recruitment signal, with expected recruitment would be at this horizontal line of 0, and anything below that would be considered low recruitment. And what really jumps out, I think, from these slides is this recent years where all of the recruitment estimates are below what we would expect to see given the current spawning biomass in the population. And so something is going on here, and we saw this and thought, well, we really need to try to understand what's happening, and that's sort of the origin of this project. And a first step was to look at correlations among the assessed species. And so what this slide is showing is this red box is showing, here's some of the species that are showing similar trends. And these are all the ones I just showed in the previous slide from the stock assessments. And then, but also over time, we see that the recent years are more similar to each other than they were in the past. So the stock assessments here or these analyses were terminated in the year 2015 because the last year we had that was common across all of these stocks would have been 2014. But we can see from this slide that it's these recent years, starting around 2010, that the low recruitment started for these particular species. And so I think the take home from this analysis is that this really is a multi-species issue, and it appears to have started around 2010. So we often examine and consider this issue on a species by species basis because that's how we're doing assessments, and that's a lot of times how the managers are taking the information from the assessments and acting on them. But this really is a multi-species issue, and we need to think about it that way. So for this project, we tried to devise hypotheses for what might explain the low recruitment in recent years. And here are 5 of them that we've spent some time thinking about. The first one is that maybe this is just a sampling artifact, that there's something in the data that's not quite right, and it's driving us to analyze the data in a way that we're getting this potentially false signal. So maybe it's not a problem at all, but it's just something in the data. The second one is that recruitment overfishing is occurring, and recruitment overfishing is when fishing happens to such a high degree that it drives down the spawning biomass that we would see a response in recruitment. And so that's a sort of the fishing effect on recruitment. A third consideration was that it's sperm limitation on protogynous fishes, because we noticed that most, not all, but most of the species that we see this low recruitment pattern in are protogynous fishes. So they're changing. At some point in their life, they change from females to males. And we already know that protogynous fishes can be more vulnerable to fishing than other fishes that aren't protogynous. A fourth hypothesis that we considered was depredation. We've heard from fishermen and others that when fish are released, that they're being gobbled up, or just in general that the ecosystem is such that there's a lot of predators out there that may be eating the young fish of these particular low recruitment species. And then the fifth consideration is an environmental effect. So I'm going to talk about the first 4, and then Anna is going to take over and discuss the environmental effect. Starting with the sampling artifact. So the one reason we consider this at all is that SAFMC, the fishery independent sampling that happens in the South Atlantic region, has expanded geographically over time. And so we wondered if this might create a false signal of decline. And so the expansion has largely been southward to cover more area in Florida, starting around 2010, 2011. But also more recently, starting around 2014, some expansion northward to cover more areas in North Carolina. So to examine this, and this was work that Brendan largely took the lead on, that it compared the indices of abundance that would be generated from the SAFMC data using the areas that were traditionally restricted, or the traditional sampling areas before the area expansion, and we compared those indices to those that would be developed using the full area. And we also examined the lengths that came from those data to see if this core region that had historically been sampled had differences in the indices and the length composition data they would see from the full area. And so the bottom line here is that we didn't really find any evidence of this. The indices really appear similar for most of the species that we examined, and I'll show an example on the next slide. And the patterns in the composition data, the length composition data, were really consistent with fishery dependent sources, and we already know that those really haven't undergone any geographic changes in sampling. So we don't think that there really is a sampling artifact that's occurring here. Perhaps there's more analyses that we could do to look into this, but at least the lines of investigation that we've taken so far really don't show any evidence for a sampling artifact. And I'll show you what I mean by that. Is that, so here's an index, index of abundance on this top panel showing relative abundance over time from the full surface area. That's the curve that's in black, solid black. And then also from the original core areas, which is mostly Georgia, South Carolina, and that's in red. And we can see that although there's some differences in a few years, the overall pattern is very, very similar. And then the bottom plot is showing the length compositions that we would get from the full area, which is in red, versus the truncated area, meaning Georgia, South Carolina, which is in blue. And again, we can see that there's maybe some differences. There's a little bit of a wider spread in the distribution when we have the full area, but the distributions are very similar. Not enough difference here that would lead us to believe that red porgy, the decline in red porgy recruitment is due to differences in these composition data. Similar for black sea bass, these are showing the same things, that indices of abundance in the top panel. And again, there's some year-to-year differences, but the overall pattern trends are very similar. And in the bottom panel, again, some differences in the compositions from year to year, but the overall patterns are very similar between the full and truncated areas. So we don't think that this is what's leading us to observe low recruitment in the assessments. The next hypothesis that we tackled was recruitment overfishing. And again, this is when fishing occurs to a high enough degree that it drives down spawning biomass, and then that leads to lower recruitment. And so just to repeat what I just said, this basically fishing goes up, that drives down the spawners, and then that in turn has an effect on the recruitment. But what it implies is that there's this order of events where fishing first goes up, and then spawners responds, and then recruitment responds. And so we can ask if we see that order in the assessment output. And so we investigated that question in a number of ways, 5 of them listed here. I'm going to talk about the first 3, which is just visual inspection of the time series, recruitment per recruits per spawner analysis, and then evidence from the fishery independent data. A couple of other analyses that we tackled were change point analysis and derivative analysis. I'm not going to talk about those, but we have that output if somebody is interested in discussing those approaches. But they basically show the same thing as the other approaches. They're all very consistent with each other. And so this is the visual inspection. And what we're looking at here, it's just take the top left panel. This is for scamp. And what's shown is spawning biomass in this purple color with the circles. And then the estimated fishing rate, which is I don't know what color that is, sort of a bluish green color. And then the recruitment pattern, which is shown in yellow. And so if recruitment overfishing were occurring, what we should expect to see would be that F goes up, spawning biomass then declines, and then recruitment declines after that. That's that order of events. But we don't really see that here. What we first see is that there's this big drop in recruitment, and along with that, actually the F is going up, but but that the spawning biomass is actually dropping a little bit after the recruitment. So there's this delayed effect. It's more that recruitment is driving spawning biomass than the other way around. And throughout this time series, F is somewhat stable, so it doesn't appear to be driving either one of them. We have GAG on the right panel. It's showing the same things. Here we do have F increasing over time, and spawning biomass and recruitment are going down over time. It doesn't necessarily have this order of events, but this is a case that's somewhat ambiguous when we look at it visually, because the SSB, the spawning biomass and recruitment, are sort of going down in tandem. So that could occur, and maybe the fishing rate is having an effect on that. So this is sort of an ambiguous case, I would say, just by visual inspection. Bottom left shows black sea bass. And again, we don't see that order of events that we'd be looking for. In fact, recruitment drops here starting around 2010, and spawning biomass follows. So it's, in this case, again, recruitment seems to be driving spawning biomass, not the other way around. And similar for, this is red grouper on the lower right panel, where we get the drop in recruitment, and then a little bit of a delay, but a drop in the spawning biomass later. But what we're not seeing is this rise in F followed by a drop in spawning biomass followed by a drop in recruitment. That would be the signature of recruitment overfishing. Another analysis that we tackled for recruitment overfishing was the concept of recruits per spawner. And what I'm showing here are simulated data, just to get an idea of what we would expect to see if recruitment overfishing were occurring. And this top left panel is showing the spawning biomass, and I've, in this simulation, have manipulated the fishing rate to be high at first, and so it drives the spawning biomass down. And then the fishing rate is less as time goes on towards the end of this time series, and the spawning biomass recovers. And the color coding is just to signify time, so that we go over to this next panel, which is showing recruits per spawner over time. We have the expected recruits per spawner is this dashed line. And if we're following time, it starts, recruits per spawner starts high, and it just jumps around, sometimes higher, sometimes lower from the expected value. And then it reaches a peak value at the lowest spawning biomass. So that would be that green area sort of in the around time 30. And then the spawning biomass starts to increase again. Recruits per spawner decreases because of density dependence. But the key here is that the simulated realized recruits per spawner really just bounces around the expected value. It can go up or down, and F can drive spawning biomass up and down, and recruits would then follow, and recruits per spawner would follow. But the key again is that it's just fluctuating around the expected line. And here I had low sigma R, which means the variability in the recruitment is somewhat low, and so we don't see a lot of bouncing around. But if I were to increase the recruitment variability, we would still see the same thing. It would just be noisier. We'd see a lot more bounciness of the recruits per spawner, but it would still be centered on the expected value. So this is demonstrating recruitment overfishing in a simulated context. And then we can ask the question, well, what do we actually see from the assessments? And so I've plotted all of the species here, but I'm just going to focus on a few of them. This first one is red porgy. And so these are similar plots to what we just looked at, where over time, the spawning biomass decreases, and then it increased, but then it decreased again. And so when we look over it to the right of that, recruits per spawner over time, we see some bouncing around the expected value, but for this ending time period where we see this sharp drop in SSB, we're getting lower than expected recruits per spawner. So it's not fluctuating really around the expected value. It's much lower than the expected value. And we see the same thing with black sea bass. It's the SSB spawning biomass is pretty flat. It increased a little bit, but then we see this sharp drop. And again, if we look at recruits per spawner, it fluctuates around the expected value for some of the time series, but for the end of the time series, we're getting much lower than expected recruits per spawner. Here's a few more examples. Red snapper I threw in, not because it's showing low recruitment. In fact, it's showing the opposite, and it demonstrates that opposite pattern in recruits per spawner. So here we just have an increase in spawning biomass starting from 1990. And if we look at the recruits per spawner, during that increasing phase, we're seeing a lot higher than expected recruits per spawner. So that's interesting. The next example is red grouper, and again, that similar pattern where during this decreasing phase of spawning biomass, we're seeing lower than expected recruits per spawner. Earlier in the time series, where the spawning biomass is increasing, it's arguably really bouncing around the expected value, but it's these last 10 to 15 years where we're getting lower than expected recruits per spawner. Similar story with gag. That's this bottom example. During the decreasing phase of spawning biomass, the recruits per spawner is much lower than the expected recruits per spawner. Two more quick examples. Snowy grouper. In this case, it's interesting because the spawning biomass is actually going up, but we are still seeing some recruitment failure, and the expected recruits per spawner at the end of the time series is the realized recruits per spawner is lower than the expected. And then this bottom example is scamp grouper, and that's that similar pattern where at the end of the time series, where we see the decrease, that the expected, the realized recruits per spawner is lower than the expected. So this is inconsistent with recruitment overfishing. The third line of evidence on recruitment overfishing that I wanted to present was the evidence that we see from the fishery independent sampling from surfs. And so for the declining stocks, what the recruitment overfishing should coincide with decreases in mean size or age. That's the prediction that we would expect to see if recruitment overfishing were happening. And if it were recruitment failure, we should see the opposite, where there's fewer of the smaller fish and younger fish. So it actually shows maybe an increase in mean size or age. So we examine the trends reports from the South Atlantic to see what those data indicated. And here's an example of black sea bass on the left. And so the relative abundance, the index of abundance, again, is showing this big decrease at the end. And so if recruitment overfishing were occurring, we would expect to see mean size decreasing over that time period. But what's shown in the bottom panel, this is total length, distributions of total length over time with the mean in the red line. We don't really see that decrease in average size at all. In fact, maybe there's some increase. With red grouper, that's the right two panels, decrease in the relative abundance at near the end of the time series. And again, we're not seeing that average size decrease that would be the signature of recruitment overfishing. It's either stable, potentially increasing from 2005, but mostly just pretty stable, noisy. Similar for scamp on the left, decreasing abundance. And if we look at the mean size below that, it's certainly not decreasing along with the decrease in abundance. And red porgy on the right, the index of abundance is decreasing. And again, we do not see a coinciding decrease in average length, potentially an increase. So we don't really see evidence of recruitment overfishing from these trends reports. And it was those types of analyses are the lines of evidence that we would use to suggest that recruitment failures have occurred for species that we don't have assessments on. So some of the ones I listed earlier in the talk that there's not a large fishery for that have demonstrated these patterns of decreasing index of abundance, but stable or increasing mean size would be more of a signature of recruitment failure than of recruitment overfishing. This is a paper that was published, a plot from a paper that was published a few years ago that looked at a number of things, but the bit that I wanted to highlight was these trends that we see from targeted species that from the fishery, number of individuals or indices of abundance, essentially, over time for the targeted species, which if you look at the conglomerate, it doesn't really show that decrease. So this includes all of the species that were targeted, not just the low recruitment species. But then if you look at the non-targeted species, you do see this decrease in abundance. And so the fact that we see this decrease for non-targeted species implies that it's something other than fishing that's driving this decline. So we think that this is not really consistent with what we'd see if recruitment overfishing were the issue. So the next hypothesis that we considered was sperm limitation of protogynous fishes, because most of these stocks are protogynous. And we don't have a lot of analysis here other than that the usual mechanism we might think of for fishing driving down a protogynous stock is that the fishing rate goes up, and because the individuals during the course of their life switch from females to males, and because selectivity of fishing tends to capture older or larger individuals more preferentially than the younger, smaller individuals, that this causes a higher fishing rate on the males than on the females. And so as fishing goes up, the number of males relative to females would decline. And if there's not enough males to fertilize all of the eggs, then this leads to decreased fertilization rates, and then that itself could lead to low recruitment. But the thing that we recognize is that this is actually just a special case of recruitment overfishing. And based on the previous analyses, we already really don't suspect that that's the problem here, at least over the last 15 years. It doesn't appear to be the problem. And so we don't think that sperm limitation caused by fishing is what's the driver here. There may be other mechanisms related to protogeny that we haven't investigated or that we can't rule out. So some thoughts that we've had would be something related to, say, environmental degradation or pollution that is disrupting the reproduction of protogynous fishes, particularly, that whatever that might be wouldn't have the same effect on fishes that aren't protogynous. But again, we haven't investigated anything along those lines, and at this point it's just sort of speculation. The next bit that we thought about was depredation, and a few species or species groups that have been discussed are sharks, potentially red snapper, because they are increasing in abundance over this time period. And so we've heard the idea thrown out that, well, maybe red snapper are eating all the black sea bass, for example. or lionfish that have invaded the South Atlantic and are populous now. So one thing we notice is that we just, if this is happening, we don't really have a good explanation for why these generalist predators would prefer to eat the suite of species that we're talking about here. Why do they like groupers and not snappers? And sort of the converse of that is, why would they appear to preferentially avoid other species such as snappers, tomtate, and grunt, which have generally been increasing in abundance over recent time? But if we look a little more closely at, say, red snapper, this is, the plots here are from the trends report, and where species are most abundant, or where they're most observed from the fishery independent sampling. And this first plot on the top shows red snapper. The red coloring is where high abundances have been observed, and the blue is where low abundances have been observed. And you can see here that red snapper tend to be off the coast of northeast of Florida and Georgia. And so if we look at some of the other species, say the next plot over on the top, this is red porgy. Well, their high abundances are generally off of the Carolinas. And so we wouldn't expect red snapper to have such a large effect on red porgy in terms of predation, just because the spatial overlap, although there is some, is probably not significant enough to drive the recruitment down to the low levels that we've observed in the assessments. And similar for this the bottom panels here show scamp on the left, red grouper in the middle, and then the stenatomus species in the which is not one that we've done stock assessments on, but also shows signals of low recruitment from the trends report. In all of these cases, their main areas of abundance, highest areas of highest abundance, don't overlap well with red snapper. So it seems unlikely that red snapper are driving down the recruitment of these species. And also, we looked at some of the ecosystem modeling that was done led by Lauren Gentry. And this was from a presentation that was given to the South Atlantic Fishery Management Council in December of 2021. And from those analyses, it didn't appear that red snapper would cause significant declines in other species. In fact, is most likely to be less than 5% for those species. And part of the reason that they identified for that is that red snapper is a generalist predator. So if a prey item becomes in low abundance, then they can switch prey. And they have really diverse diets, and not just of fish, but they eat a lot of crustaceans and other invertebrates. We thought about lionfish also, and these plots are from a recently published paper by Finch et al. That shows in these top panels the relative abundance. And similar to red snapper, you can see that the area of highest abundance is off of northeast Florida. And so again, the spatial overlap with species that are showing low recruitment doesn't match. And another bit that was modeled was the relative abundance and probability of occurrence. And you can see that it's really not increasing anymore. It was increasing for a while, but over the last few years, it's decreased or stabilized. And so if lionfish were the culprit here, we might see a response in recruitment where it might start to rebound if lionfish predation were the issue. And again, we don't think that necessarily is the driving reason here for the low recruitment. And so that is what we have for the first 4 hypotheses. At this point, Anna is going to take over and present what we've done on environmental effects. And I'm just going to hit slide forward whenever Anna says that I should. And we can come back to questions on anything previous at the end.
Hi, good afternoon. Thank you, Kyle. Thank you, Chip, for the introduction and the opportunity. So I'm going to talk a little bit about our ongoing work with the environmental effect. If you please, Kyle, can press forward. We're going to start is talking a little bit about the recruitment drivers. So as Kyle said, it's important to understand the spawning, biomass, and recruitment relationship. So there is a new paper this year that reviews this relationship between spawning biomass and recruitment, and they categorize stocks into 3 categories of recruitment drivers: spawning, biomass driven, environmental driven, or edge stocks, which have contribution of both over time, different, sometimes driven by SSP, sometimes driven by environmental effects. So we apply the same methodology to our region. So stocks that were significantly correlated between spawning biomass and recruitment, the recruitment was considered dependent on the spawning biomass. And stocks that did not have a strong correlation between both would be the cases where the environment is playing a role. And for some stocks, maybe it's a combination of both, as Kyle has gone over, like the graph showing that case. So if we go to the next slide, please, Kyle. Kyle did this analysis using the output from the stock assessment, and we've seen that we applying the same analysis to our species. Out of the 12 species we consider, 5 they were considered to be that edge case. That was the special case where both the spawning biomass and environment had an effect on recruitment. And we can see here these 5 species. Interesting is 5 of the species we've seen the trend of low recruitment. So red pork, black sea bass, gag groupers, snowy grouper, and scamp. Out of the other species, 7, all the other 7 did not have a strong correlation with spawning biomass, and so they were considered to be environmentally driven recruitment. And many of them are not decaying, they are increasing their recruitment, and even in this case, they have this relationship that is environmentally driven. So what could be influencing this, then what can be influencing our species? One clue we may have, please the next slide, is from this table that was compiled by Brandon, that he compiles spawning seasons of a variety of species in our region. And we can see that all species that we have decreased abundance, where we have evidence of low recruitment, are species where the peak spawning occurs between February and April, about what we say winter spawners or early spring spawners, with maybe a few exceptions like sand perch that goes more into the summer spawning. But the majority of them have the peak spawning in February, March, while other species that are showing stable abundance trends, or they show increasing trends, they have no signs presently of low recruitment, that are show the species in green here, including Amaco jack, land snapper, red snapper, vermilion snapper, white grunt, and mouton snapper and gray snapper. They are summer spawners, or with especially their peak spawning between June, July. So that shows us that maybe these external environmental factors with some seasonal patterns could be affecting the recruitment in our region. Another piece of evidence, please, next slide, Kyle, please. Next. Kaya?
Uh-oh. I hope he's not locked up.
Yeah.
Anna, is your next slide the, let me see, on changing ocean conditions?
Correct. Can you, sorry, I don't have the presentation downloaded, so be hard to share my
Yeah,
screen.
I'm not positive this is the most recent one, but I'll show my screen. Hold on one second.
It should be mostly okay. There's just a couple. Yes, thank you, Chip.
Sure.
So yeah, so another piece of this puzzle comes from the last environmental status report for the South Atlantic that compiled in 2022, who was led by Kevin Craig. And one thing which this report shows, our region has an increased sea surface temperature, particularly after 2013. We can see in the first figure this trend of it shows the anomaly, the variability of the sea surface temperature over the mean over time. And we can see the last years, you have a trend of increasing temperatures. It has at the same time about, if our region, you're gonna have like two types of upwelling. Upwelling is the transport of this deep, cold, nutrient-rich water into the continental shelf. You're gonna have the upwelling that is driven by the coastal upwelling, the wind patterns influencing, and the large scale upwelling that is gonna be driven by the Gulf Stream variability. So we can see this upwelling here, we have an index showing how much of this nutrient-rich water is being carried into the shelf is measured at a coastal point. Actually, this is the data, and it shows a decrease in this upwelling intensity. Over the same amount of time, we have an increase in the sea surface temperature. Another piece of this puzzle is the position of the Gulf Stream, because the Gulf Stream also drives some of this variability. And at surface, we can see in these recent years, just more onshore position of the Gulf Stream, which could make it not contributing to upwelling in the region. We have then all these factors affecting nutrient dynamics. If we look at the chlorophyll for the same time period, average for our entire region over the shelf, continental shelf area, we would expect a decrease into this chlorophyll A amount, because we're going to have higher temperatures, you're going to have less nutrient-rich water being transported into the shelf. But we don't see this pattern when we average the conditions over the whole region. There's some years that you do have a smaller chlorophyll A. There's a lot of variability, but there's a certain increase in this chlorophyll A in other months. And the top figure, if we look here over the trends of chlorophyll A in our region of this time period, you can see that there is a lot of spatial variability. That means there's just areas, the coastal areas in red, which have an increasing trend of chlorophyll. And they're going to be more productive. So they're probably the areas that are related to these peaks of chlorophyll, while other areas show a negative trend, which is areas that chlorophyll you might be getting less chlorophyll in the most recent years. So while there is some suggestion here that we could be having conditions that could lead to lower productivity that could affect our recruitment success, it was not a very straightforward picture. So we are exploring this. Next slide, please. Thank you. Exploring this into trying to get better the spatial and temporal patterns of this recruitment. So we are doing, we're approaching this by two angles. First, we are exploring how exploring explanations are hypotheses related to the environment. So if we know, we think there is reduced productivity, we're trying to understand if that could be a mechanism that is leading to lower recruitment by understanding if there is more lower stratification of the water column because there's more heat. So the whole water column is heated, and then you're going to have less like cold rich water being transported in the shelf. So it's going to be less productivity, less chance of having food for the recruits or for the stocks themselves. And the others just look at what is how they varying, how these environmental variables are varying over spatial and temporal scales that are important to our stocks. How is their variability over the places where both adults and recruits are, and over the seasons of spawning, the important seasons for them. One issue with our region is that we have a data limitation. We have a relative low coverage of sampling for oceanographic variables. So here we have like these little figures there. They show data points on the left from our surveys, from SURFs, MyMap surveys across 30 years, and on the right we have CTD, that is the cast, the instrumentation to get temperature, salinity over the same time period from publicly available data sets that are from other researcher institutes or academic partners. So in total, we have about 7,000 points over 30 years, which is not enough for us to really capture these changes over space and time. So to overcome this limitation, we use oceanographic models outputs to have data that is continuous over space and time. So next slide, please. So we are using the Couple Northwest Atlantic Prediction System, that is short for SYNAPSE, which is developed by Roy Hay at North Carolina State University and his team. This is a figure of the model domain, so it covers the whole Western Atlantic, Gulf of Mexico, the Caribbean. And it's a very long model in high resolution, which you would say, because it has the grid size is about 4 kilometers. So each 4 kilometers, we have a grid that we have a data point there. It has a very good also resolution of the vertical in the ocean, and it comprises from 1993 and is run to present day. We have data until last year. You have daily fields. We are using sea surface temperature, sea surface height, which are the deviation of the anomaly from like the medium of the height, the mixed layer depth, the surface salinity, and bottom temperature in our analysis. We also use chlorophyll weight as a proxy for primary productivity from 2 different satellite products. One is just the Moody's Aqua is a satellite. The other is a European, the Copernicus Globe Color, which they compile every satellite data available for chlorophyll weight into a merged product. So we use both of them for comparisons, and we are using them all these data sets. We are particularly interested in understanding the seasonal scales of variability. So most of them we are calculating them by seasonal scales to understand these differences between species that are spawning in winters and versus species in the summer. So the next slide, please. So here we have on the left panel, we have anomalies of just some of this model variables, the oceanographic model. So the first one, they are for winter. So is the anomaly is just the difference over the mean for the time period. And this is just each point there is one winter over this 30-year time period. So the first one is the winter, the anomaly of the sea surface height. The second one is the bottom temperature. And the third one is the sea surface temperature anomaly. And we can see for the three variables, the anomaly is we have an increase over the last more than 10 years, since about 2012, 2013. You have an increase in sea surface height anomaly, so you have higher sea levels, you have higher bottom temperature, and you have higher sea surface temperature during the winter. We're gonna talk a little bit more, but it's very straightforward to understand how temperature can be affecting our species, because temperature itself might drive species distribution or can influence the prey, the productivity. But one thing is interesting is like, what is the sea surface height doing here, right? So in our region, sea surface height, the sea level, that anomaly there is mainly driven by the Gulf Stream, because the Gulf Stream changes the sea level. So the movement of the Gulf Stream onshore, offshore itself can change, help change the patterns of the sea surface anomaly. So that is acting kind of as a proxy for the Gulf Stream variability over this time period. So these anomalies are taken from Cape Hatteras to Cape Canaveral from 0 to 300 meters depth. So after we calculate the anomalies for all the seasons, we have all the variables in all the seasons, the 6 variables I mentioned before. We correlate them to our recruitment deviations from the recruitment time series of the species. And we can see is in this big matrix here. So the variables that are mostly correlated with both the ones that the species showing low trends of recruitment and the species not showing low trends of recruitment are just variables we have plotted here. The winter sea surface height anomaly, the winter bottom temperature anomaly, and the winter sea surface temperature anomaly. We sliced these data sets in different ways. We did some other statistical analysis to look at the not the anomalies themselves, but trying to look at the major modes of variability of this data set using statistical analysis. And when we try to separate like these oscillations, like which is how they changing according to other factors in the system, decadal scales and things like that. These variables still are the highest correlated. So even when we did different treatments, statistical analysis to get these anomalies over time, not just getting an average, the deviation over like the average, we still got that these 3 variables at these time periods were the most correlated to our recruitment. The next slide, please. So as I mentioned, the Gulf Stream seems to be, it could be an important piece of our puzzle, both because it drives variability in the system and it can drive the upwelling, but also because we saw that the SSH, the sea surface height, is correlated to the recruitment deviation of several species. And so here we have these graphs in the left. They represent the oscillation of the Gulf Stream over time. If the Gulf Stream is being more inshore, or if the Gulf Stream, in average for a whole area, is more offshore. So we have the calculation of the Gulf Stream position for every day, which is the black line in the back of the graphs. The top graph showed the oscillation of the Gulf Stream at surface. So anything that is positive the current is more offshore. Everything that is negative, the current's more inshore than the average. And because this is a very wide and deep current, we not just look at the surface, but we also look at subsurface, which subsurface here is a depth, is a 200 meters depth. So we look how the center of the Gulf Stream at 200 meters is changing to offshore or inshore. So we can see on the top left graph, the surface one, that the position of the Gulf Stream at surface doesn't seem to change much over time. It seems pretty regular. If nothing, there is a little significant trend of a small tendency to be offshore in more recent years. But we can see that at subsurface, there is a clear increase of the tendency of the Gulf Stream to be located onshore, which we call the intrusion of this subsurface water, which could be a representative of upwelling, because you have in these colder waters that are flowing down, being pushed into the shelf. These Gulf Stream intrusions, they historically occur during summer months, but when we separate these patterns that we see here by seasons, we see that there is evidence of frequent winter intrusions, particularly since 2010. If we do the correlation analysis of these subsurface patterns of the Gulf Stream, so how much is coming inshore or offshore, separate that by seasons, and correlate the seasons to our recruitment deviations like we did in the slide previously, we see that there is a lot of the winter movement of the Gulf Stream at depth is correlated to a lot of species. As expected, it's a decay, like a decay, which would be the current coming more onshore in the winter. It's positively related to species that are having low recruitment. So current coming onshore relates to low recruitment, and inversely correlated to species showing increasing recruitment, such as red snapper, vermilion snapper. So we still need to explore here what is the role of the subsurface intrusion if this entrance of the Gulf Stream at depth can be changing maybe productivity, primary productivity. Maybe that could be a hypothesis we can work with, is the match-mismatch hypothesis. So maybe is increasing productivity at a time that is not useful for the winter species, but is going to be beneficial for the summer species. So that is something we're still ongoing. So next slide, please. So our results are overall, Kyle's back. I don't know if you wanted to present that part, but they're still preliminary. We have not a smoking gun on what's driving the low recruitment. As Kyle presented, there was no evidence for sampling artifacts for recruitment, overfishing, protogeny, or depredation. And our main current focus is on the environmental drivers. Recruitment overfishing also does not appear to be the primary driver of low recruitment. In recent years, at least, it could still play a role as suppressant of recruitment, and may have been an earlier driver in the 70s or 80s when there was declining abundance of multiple snapper grouper species. And the main takeaways of the environmental drivers is, please, the next slide, Chip. Thank you. Is that we have a significant correlation between winter variables, especially surface temperature and bottom temperature, with recruitment deviations. We have more, the Gulf Stream at depth is coming into the shelf, particularly at winter, and is significant for correlated with several species recruitment. We need to explore now relationships between recruitment, is Gulf Stream variability, and chlorophyll way. explore if there is a role of the wind and changes in the stratification of the water column with higher heat, and consider the combined effects of the environmental covariates instead of working with them all separated. One thing that is also ongoing, and we're going to start, is looking at how the transport of recruits actually simulate transport of recruitment to see if there is any environmental changes on the Gulf Stream position or eddies or other features that could be leading to pulses of recruitment or actually transporting recruits offshore and contributing to the patterns we observe. So that is it. I think we have a thank you slide and open for questions.
All right, thank you both. I'm going to bring back up the other slide real quick. Just reiterate how to operate the webinar. You can type a question into the question box if you don't want to ask your question aloud. You can also raise your hand, click on this icon that is a hand-drawn hand. If it's green, that means your hand is lowered. If it's red, that means your hand is raised. And once we've seen your hand is raised, we'll notify you that you're the next speaker by clicking on the webinar to recognize you. If your microphone is red, that means it's still muted on your side. Just click on that red microphone, it will turn green to indicate that we'll be able to hear you. With that, I'm happy to take any questions. And this is just another presentation of what the dialog box might look like in different apps on different platforms. So I'm not seeing any hands or questions right now. There we go, George.
Yeah, can you hear me?
Yes, we can.
Yes, we can. Okay, I was wondering in the predation work, if it's possible that the predators and the prey are life history stages. They're not being sampled by the SAFMC program or being targeted by the fishermen. So it may be that juvenile snappers or groupers that are not vulnerable to the surf scare are targeted by fishermen, are actually the ones that might be preying on early life history stages that are also not sampled by surfs or targeted by the fishermen. You know what I mean?
Yeah, that's a good question. I guess two examples might be gag or black sea bass that have the estuarine dependent phase that wouldn't be sampled by surfs. So at least for those two species, it's possible that they're being preyed on during that juvenile phase by other species that aren't included in our analysis. So we haven't done any analysis on that topic to address your question, but other than to say that, yeah, they think that at least for some of these species that might be possible.
All right, we have a question that's been typed in the question box. Do enough of the low recruitment species share a nursery habitat type to justify exploring low recruitment drivers specific to inshore habitats?
I think the two that I named, black sea bass and gag, are the ones that stand out from our list. The others, I don't think they're juveniles phases are inshore. I guess that's not a direct answer to your question of whether it justifies doing the analysis. But I think it's unlikely to be an explanation for all of the species together, but it could play a role for at least for a couple of the species.
Go ahead, Judd.
Thanks, Chip. Thanks, Kyle and Anna. Great presentation. We've seen that some of these environmental drivers are causing shifts in like species distributions. Have you talked to anybody else in other regions, the Mid-Atlantic, the Gulf, New England, to see if anyone's working on some similar studies to see if they have any recruitment failures in some of their managed stocks or non-managed stocks?
I think maybe black sea bass is one that's gotten a lot of attention about having a distributional shift northward. I think that's potentially true. It's also might be convoluted with lower abundances in the southern regions because of higher fishing rates, and so the models that are typically used for that, usually it's a vast model, and it might not be able to really distinguish shifts from just relative abundance changes by area for other reasons. But certainly black sea bass is one, and species that aren't necessarily in both regions that have been investigated in the Northeast. There's a paper published recently on declining population trends. I don't know that it was explicitly related to recruitment failure, but they were able to tie environmental conditions, especially temperature increases, with some declining patterns in abundances.
Are you seeing any changes in the timing of when the different species are recruiting to their areas? I know it might be difficult, but maybe something like MRIP data, even though it's reported on waves, you might be able to see if something like black sea bass are coming into the estuaries at a later time, or another data set. Have you guys been able to look at the timing of recruitment? I know with some of the shrimp work I'm doing right now, they're seeing shrimp are coming, some of the species of shrimp seem to be coming into the estuaries at a later time period.
That's a really interesting question. I don't think we have the resolution and the data that we have available, or at least that we've analyzed to really address that question. But I imagine there would be some state data that we could look at least for the estuarine dependent species, to see if there's changes in the timing of when those species are showing up inshore, the juveniles are showing up inshore.
Go ahead, Marcel.
Thank you. Can you guys hear me?
Yes.
Thanks. Very, very interesting work. Similar to the previous question, and I know there's some efforts underway. Have you guys looked at potential changes in reproductive parameters, in particular, for instance, changes in the length or the beginning of the spawning season for some of these species? Thanks.
That's something we need to investigate more. We've looked a little bit into body condition, changing in body condition of red porgy over time. Nothing, no smoking gun there, but those types of things I think we could look at more in depth, investigating changes in life history characteristics over time. So I guess the short answer is we haven't really done a lot of that, Marcel, but I think there's some room to do that.
Yeah, I don't think it's a cause for, well, maybe if there's a shorter reproductive season that may affect the recruitment, but I was more thinking about the mechanism between the environmental factors and the recruitment issue. So thanks, I appreciate it.
Yeah, I mean, part of the challenge with addressing that question is that the fishery independent sampling is mostly through the summer, so we don't have a lot of samples, other than special studies, for example, during the spawning seasons that happen in the winter or spring. So we need more funding so that SAFMC can operate throughout the year.
I agree. Thanks, Carl.
Well, I don't think funding will get you there, unfortunately. I think you have other issues that will prevent you from sampling throughout the year. One question I had based on when you were talking about looking at the condition index, were you trying to look at competition among species to see if the fish were, I guess, leaner and therefore they might be in competition with another species? Is that what you were looking at with that?
No, it was more, I mean, that could be part of that, but we hadn't gotten that far with it. It was more just looking at body condition on an annual basis, average body condition on an annual basis, to see if there's any patterns that emerged. And that was, again, just for red porgy. And Nikolai was leading the charge on that analysis.
Any other questions? All right, I'll give it another 10 seconds. Go ahead and raise your hand or type a question into the question box. Well, seeing no more questions, we're going to let you off the hook early today. Kyle and Anna, thank you so much for a really great presentation and for doing the work. I think it's extremely valuable to see these kind of insights into the fishery populations offshore or fishery populations in general, and trying to understand the impact of climate and other factors on the population. So we greatly appreciate it.
Thanks everybody for the interesting discussions.
Yeah,
And then next, oh, sorry, Anna.
Sorry, thank you for the opportunity to share. Yes.
And next month on August 13th, we are going to have our next seminar series. Matt Damiano with National Marine Fisheries Service is going to be talking about dolphin. And so we look forward to having everyone back for that presentation next month. Thank you for attending.